Yeah, this was a talk given in the Clinical Science Symposium and essentially brought three oral presentations from different disease types together, one from myeloma, one from lung cancer, and one from breast cancer. I essentially gave the foundational lecture of where we are in terms of antibody drug conjugates. So essentially, I think this is hard to imagine that the first antibody drug conjugate was approved 24 years ago. And this was gemtuzumab ozogamicin. This is in the liquid tumors. It wasn't until 13 years later in 2013 that we actually had the first antibody drug conjugate in solid tumors. And that was trastuzumab emtansine, or TDM1, for highly HER2 expressing breast cancer. One of the big trends we've seen over the past several years, development really exploded for antibody drug conjugates around 2019. We now have 6 that are FDA approved for solid tumors, 6 that are approved for liquid tumors, for a total of 12 approved agents that are ADCs in oncology. We've seen that at the beginning, all of these agents had to highly express the target. So HER2 for TDM1 or CD33 for a liquid tumor target, for example. Now we have drugs like sacituzumab govitecan, which is a TROP2 targeting antibody drug conjugate in breast cancer, where we don't even have to test for TROP2. Similarly, trastuzumab deruxtecan as part of DB04, and now DB06 was presented by Dr. Crigliano at this year at ASCO, showing that we can go all the way down to HER2 ultra low, meaning even patients that have IHC 0 expression. So what we're learning is with differences in not only the antibody, but also really that linker and the payload, that sometimes high expression of the antibody isn't needed anymore. And that's really encouraging. One, because it's available for more patients. We're not having to test. And testing can be a little bit problematic because expression of these antigens really vary depending on where we put our needle in and what tumor lesion in the body. And we're also seeing, good tolerability with these agents. Really across the board, they've all beaten chemotherapy, which is incredibly exciting. So we spent a little bit of time talking about what's on the horizon, really figuring out how much antigen expression matters, talking about new payloads, maybe even bispecifics, where instead of just having one antibody, it targets 2 different antigens on the cancer cell. You can imagine that this could cut down in terms of resistance. We talked about some tricks like payload masking, where we essentially protect the antibody drug conjugate from having any activity. And then when it's in the tumor microenvironment, that mask is cleaved off and the antibody drug conjugate can go to work. That's going to translate into more tolerability. And so there are really a lot of antibody drug conjugates now in development, both for solid and liquid. And I really think that these are going to revolutionize oncology care. One big question we have is terms of resistance and how to sequence these agents. We know we can see resistance to not only the antibody, but also the payload. And so we think we may have better activity when we both switch the antigen target and switch the payload. And so there are a variety of drugs being developed in this space.